Hello, everybody. Welcome to our CDCR seminar, where we have our very own Dr. Sheila Singh presenting today. She's no stranger to anyone in the audience. I can't help but notice this is a much bigger turnout than it was for when I talked, but I understand why. No hard feelings. So Sheila, as everyone well knows, is the director of our Center for Discovery in Cancer Research. She holds a Tier 1 Senior Canada Research Chair in Human Brain Cancer Stem Cell Biology, and the founding director of the McMaster Surgeon Scientist Program. Her PhD thesis described the novel identification of a population of cancer stem cells that exclusively drive the formation of brain tumors. Since 2007, Dr. Singh's lab applies a developmental neurobiology framework to the study of brain tumor genesis. Building upon previous cell culture techniques developed for the isolation of normal neural stem cells, NSC, and applying them to brain tumors, and through development of a xenograft model to efficiently study brain tumor initiating cell BTIC activity. Dr. Singh's lab aims to understand the molecular mechanisms that govern BTIC self-renewal. She's currently studying the regulation of BTIC signaling pathways in glioblastoma, brain metastases, and childhood medulloblastoma, with an ultimate goal of selectively targeting the BTIC with appropriately tailored drug and molecular therapies, which is a place where a few of us are coming into the fold as well. Her laboratory is funded by just about everybody, by CCSRI, CIHR, TFRI, a lot of these acronyms I don't even know, CRS, the Stem Cell Network, McMaster Surgical Associates, Brain Canada, and the Boris Family Fund. She's scientific founder and prior CEO of a startup company, Empirica Therapeutics, a brain cancer therapeutics company that is seeking new data-driven and polytherapeutic treatment options for patients with glioblastoma and brain metastasis. Empirica was acquired by Century Therapeutics Incorporated in Philadelphia in June 2020, resulting in the creation of a Canadian subsidiary, Century Canada, which I'd love to have some shares in, based in the McMaster Innovation Park in Hamilton. And what's very impressive is she's done all this with only one hand. The other hand has been serving as professor of surgery and biochemistry, chief pediatric neurosurgeon at McMaster Children's Hospital, and division head of neurosurgery at Hamilton Health Sciences. So it's a pleasure to present to you, Sheila, without further ado.
Thank you, Anthony. Thank you so much for that kind introduction. And I am so thrilled to see all of you here. I took the July spot because I thought everyone would be on holiday. And I thought, oh, it's just me, and everyone's heard me speak a million times. So, but now I realize that I have been lecturing a lot over the past few years, but it's all over the place. And I rarely get the chance to speak to my own people. So I'm really, really happy to see you all here today. And that's why I chose this particular program to feature, because even though I love all three programs in the lab equally, so GBM, brain metastases, and medulloblastoma, this program has a special place in my heart because these are my actual patients. So as a pediatric neurosurgeon, I treat these children, and I usually meet them when they're very, very sick, in fact, at their sickest. And I take them through surgery as the first step of their treatment. And I don't abandon them when I finish operating on them. Then there's a long treatment course after that. And I move on to our multidisciplinary pediatric neuro-oncology clinic, which I also co-direct with Dr. Adam Fleming, our incredible pediatric neuro-oncologist. And then it's a team-based approach where all of us are working together, radiation oncologists, medical oncologists, neurosurgeons, neuropsychologists, OT, PT. It's a huge allied health team. And we work together to take care of the child as we see them through standard of care therapy, which actually lasts anywhere from 8 to 13 months of really intensive heavy chemo and radiation, and sometimes stem cell transplant if they're the right age. And then they come out of all of that, and then we cross our fingers and hope that those children don't relapse. And fortunately, I saw the 5-year survivorship for medulloblastoma increase just over the course of my training. So I've been here 17 years now, and during the course of my neurosurgery residency, I managed and treated many children with medulloblastoma at the Hospital for Sick Children in Toronto. So if I look back over the course of the last 25 years, the 5-year survivorship has actually improved overall from about 50% to 80%. And that's a remarkable shift in a field. If you think about 2 decades, what an incredible improvement. And those were with refinements of microsurgical techniques, the fact that molecular profiling of medulloblastoma hit the stage in the early 2000s and really completely reshaped the way we think about the disease and establishing molecular subgroups. And also the fact that those molecular subgroups led to at least the hope and the development of predictive and prognostic therapies. So all of that improved things greatly. But the 20 to 30% of children who relapse have absolutely no options because we throw everything but the kitchen sink at them during their standard of care. And when they emerge from that, if they do relapse, they literally are resistant to everything. And this is the most difficult problem to tackle. And so that, of course, is what my medulloblastoma team in Singh Lab is attempting to tackle. And it's a very difficult problem, but it's for some reason, and I'll explain a little more in the talk, but I feel incredibly hopeful about this field, that because we made that huge jump in survivorship over 2 decades, I feel like this is eminently a solvable problem. And I think we're going to see that even those relapsed children will have new hope in the next 2 decades as well. So today I'm going to tell you about our understanding of clonal evolution, focusing on recurrent treatment refractory medulloblastoma. I'm going to share with you some of the models that we built over the past 2 decades to try to help us further our understanding of this disease. Oh, except I need to advance. There we go. So today I'll be speaking a little bit about the medulloblastoma program, and it's not a comprehensive overview. I don't have time to share everybody's work, but you'll hear a few stories today from the medulloblastoma program. So first, I'll tell you my entire incentive for doing my PhD, because that is actually built into this program. So I was a neurosurgery resident in my 2nd year of training, and I had just come off neuropathology, and I was starting my pediatric neurosurgery rotation at the Hospital for Sick Children. And I had something that I would call an epiphany, because it really shaped the course of the rest of my career. So I met 2 young boys, and they were both named Christopher. And they were on my service, on my team, and I was taking care of these children. So taking care, when you have children on your service, you're really seeing them like every day, early in the morning, late at night, and you follow them really for months. And your rotation is 6 months, so you get a really close bond with the children and their families. So they were both named Christopher, and they both came in with headache and vomiting. Okay, and people have pointed out to me, I know this is the same MRI. This is an illustrative story. So these are not their actual MRIs, but they both came in with this large, you can see the brain tumor clearly here. So it's large, it's heterogeneous, it's horrible looking. It's actually occluding the fluid spaces in the brain, which are black. These are the fluid spaces. These are the lateral ventricles draining into the 3rd ventricle, normally draining through the cerebral aqueduct into the 4th ventricle, normally draining through the outlets, foramina, medulla, and luschka to circulate around the spinal cord in the brain. But you can see there's a big block there, right? There's a big block there because this huge tumor growing within the 4th ventricle, perhaps arising from the roof of the 4th ventricle, or maybe from the ventricular zone, is actually occluding the flow of the spinal fluid. And that's why these ventricles are 2 or 3 times bigger than they should be. And so when the child comes into emergency, they have headache and vomiting, not from the brain tumor, but from the hydrocephalus, which is acute. And then this brain tumor is causing some cerebellar problems. So the cerebellum controls fine motor balance and gait. And so these children come in broad-based ataxia, we call it, when they're falling and they're clumsy. So both Christophers came in with very identical presentations to emergency within 2 weeks of each other. And they both went on to have best possible treatment. So we rushed them both to the operating room, and they got a near total resection. So we try to remove everything except for, we might leave a few millimeters of disease left on the floor of the 4th ventricle, because that's a very important anatomical zone that you can't afford to breach. We've learned that chemo and radiation can clean up the majority of that microscopic disease we leave behind. So we do a near total resection. So both of these kids had that, and they came up with a quick section diagnosis of medulloblastoma. This is the most common childhood malignant brain tumor. It's grade 4 out of 4 grades. It's very aggressive. It's very proliferative. The Ki-67 index on the slides, even on the quick section, if they do it, it's scary. It's usually 90%. Sometimes it's close to 100%, which is terrifying of how quickly these tumors must be growing incrementally quickly. And so both of these children went on to recover from surgery, and they both were enrolled in the high-risk protocol for medulloblastoma, ACNS 0935. So after their surgery, they actually went on. Both of them were diagnosed with classical medulloblastoma. This is one of three pathological subtypes. So really, if you look at this tumor, it's identical in both children. It really, really, really looks very similar. And in fact, their treatment course is going very similarly as well. They've both gone on now to 3 chemotherapies and craniospinal irradiation, and we are caring for them through all of this. So both of the Christophers went through looking as if in every sense that we can measure this tumor. So pathological, anatomical, every measure we have of this tumor at this time, they look identical. And so it really genuinely shocked me when we provided this gold standard care to both of them, that one Christopher died 15 months post-diagnosis, and the other Christopher survived. And I followed his story well beyond my rotation, and his mother founded a charity called Brainchild with the other Christopher's mother. And so this divergent outcome really shocked me. And then I realized that no matter how much I learned about neurosurgery, I would never understand why one Christopher survived and one Christopher died unless I did a PhD. So that's when I decided to go and do the surgeon scientist program at the University of Toronto and enroll in the lab of Peter Dirks. So that's really sort of my epiphany, really came from wanting to understand more about this disease so that we could really understand why. And this is Heraclitus, and he said something, this Greek philosopher said something very wise, which is not surprising. He said, one cannot step twice in the same river. And it struck me as I was finishing my residency that every way that we assay cancer is generally static, right? You take one biopsy from the child while they're going to surgery, and we look at it under the microscope or we study it, and we get a very static impression that that's the cancer, that's what we're treating, go. And then big pharma can invest like hundreds of millions of dollars into that one biopsy to try to develop targeted therapies and so on. But we're not taking into consideration that just like the river that Heraclitus is describing, cancer is not stable, it's dynamic, and it's changing and evolving through time and through space. Just like the water flowing over your foot, if you put your foot in this river and the water is flowing over your foot, it's never the same molecules that are touching your foot at any one time. You can just say, yeah, my foot's in the water. That's the water. It's the same water, isn't it? It's not. It's always changing. And so that made me realize that when I started my own lab, my first task that I would assign myself would be to develop more dynamic models of cancer, a way to look at cancer that is not static, and a way that could not just look at it as one snapshot in time, but to model cancer through time and space. So that was what I wanted to do when I started. So I've already told you all of this about medulloblastoma in my very long preamble, so I won't belabor this point. But I also want to tell you one more thing, is that it's not only a goal to try to find better solutions for the 20 to 30% of children that relapse. It's also the fact that the 70% or 80% of children that we cure have really bad neurocognitive treatment effects from that horrible systemic toxic therapy that we give them. Because remember, chemo and radiation really does damage every cell in the body, every proliferating cell in the body. So these children are left with devastating side effects. And those have improved over time, but wouldn't it be great if we could develop a more targeted therapy that spared the normal cell populations? So the second thing that I always had in my head for when I started my own lab was to try my best to take a comparative biology approach to everything. So don't study cancer in isolation, study cancer compared to normal cell biology, and try to distinguish how cancer, especially in a childhood cancer, might actually represent development gone awry. And so that understanding can help you in two ways. You can understand how a cancer cell hijacks the normal developmental mechanisms of a normal cell. But then the second advantage of that comparative biology approach is that you can find therapeutic windows, find things that are cancer selective. By profiling both normal and cancer cells, you can find therapies that are cancer selective and that spare the normal cell populations. So those were sort of two goals that I had set for myself. And fortunately, I have a lot of really good colleagues who I trained with at the Hospital for Sick Children, along with Peter Dirks was Michael Taylor, who was 2 years ahead of me in neurosurgery residency and was kind of like a big brother figure in the lab, always mostly telling me what I shouldn't do. He never really told me what I should do. He always told me what I shouldn't do. And he was there to give me advice. And he had entered the lab of Jim Rutka. And Mike really blew out of his PhD as one of the 4 major groups in the world that established the new molecular profiling of medulloblastoma. So he actually discovered through very early genomic analyses that medulloblastoma is not 1 molecular disease, it's 4 molecular diseases. Actually, now it's 13, but it was 4 back then. So what you can see here is that Mike and others have actually done a lot of work into trying to understand not just the primary disease, but the disease as it evolves through therapy. And so there's a lot of evidence that was generated in all of these other programs to suggest that actually there is a clonal divergence that happens through therapy. There are populations, much like evolutionary biology, that are selected against by therapy, that are selected for by therapy. And what emerges after the therapeutic bottleneck has a very different phenotype than what is there at the beginning. And so this gave us a lot of information in terms of the goals of the Singh Lab. And what I had learned, of course, during my PhD was that the gold standard models for stem cell assays are patient-derived xenografts, where you take patient cells and you can transplant them into mice serially at ever-limiting dilutions to try to understand which are the actual populations that are initiating disease. So trying to find tumor-initiating cells. So armed with those models from my PhD, I brought them here to reestablish them at MAC. I didn't bring anything actually, but I brought the knowledge to set up the models here. And what you can see here is that I was very determined to try to set up a model of medulloblastoma recurrence. Because one thing I didn't mention to you is that when children with medulloblastoma recur, we're at a loss to find new therapies for them, but also we're rarely in a position to let their biology drive our treatment choices because we rarely biopsy children who have recurrent medulloblastoma. And this has really been something that has set us back because how can you have a biological understanding of the disease when you don't have a sample of it? So I rationalized that developing this mouse model, this patient-derived mouse model, we could capture that recurrent evolution to recurrence in mice and really try to understand the recurrence. And so the goal of this was really to do integrative multi-omic profiling at every stage of this model to try to understand not only the relapse, but perhaps even more informatively, this stage that just precedes relapse, which in leukemia is called minimal residual disease. And this is a stage that is not well characterized in brain tumors, but really deserves more attention because this is kind of an anticipatory profile. If you could capture transcriptomics or proteomics of this stage, imagine what you could do to anticipate and try to block the disease rather than treating it when it's already formed. So this evolutionary model of clonal evolution of medulloblastoma through therapy was the subject of my very first CIHR grant. And the nice thing about it is that it not only serves as a discovery platform, but this also serves as a preclinical testing platform. And this has enabled us through collaboration with people developing new targeted therapies for medulloblastoma to layer them on to the standard of care or to insert them somehow into this model to try to understand how that changes the clonal dynamics of what we see as well. And so here, this is some work done by our former PhD student, Dr. David Bakshinyan, who is now doing his MD at University of Toronto. And what you can see here is that he did this lovely profiling where at every stage we sacrificed mice and did some targeted proteomics. And you can see here that we're really able to understand the proteins that are really enriched at relapse. And furthermore, when we run models like this, this is good for David, but it also establishes a database for the lab. And this database can be used for many establishment of many further projects. And then you can see here the integrated multi-omics approach can't be done just by one lab or one person. And so we've had really great collaborators who have helped us collect many levels of disease, to profile the disease. And at the same time, we in Singh Lab are expert in doing functional assays. And so when we recover stem cells from every stage of this model, we're able to do functional profiling and set up in vitro and further in vivo stem cell assays to really understand functionally what's going on as well. And so all these levels of data have been really helpful to understand a relapse. And again, you can see here that it's important for us also to have visualization. And we're very lucky to have Manoj Singh in the lab, who is able to perform MRI. And this really helps us to inform about when the tumors are engrafting. This gives us information about when we can start our new treatment protocols, and then we can model the disease through time as well. And so having that anatomical parameter and the information that Manoj can provide has been really helpful to us. So you can get an idea. Rulo talked a little bit about my training in stem cell biology. And certainly, especially out of all the tumors that I study, pediatric medulloblastoma is perhaps the one to which the cancer stem cell hypothesis most likely pertains. And the reason I say that is because if you think about a disease that evolves in a child is exactly the type of person who has very large stem cell populations because their brain is actively developing. And then layer onto that the only part of the brain that continues to develop postnatally is the cerebellum. The cerebellum is actually continuing to develop until the age of 2. The window when children get medulloblastoma is between the ages of 3 and 5. So it makes a lot of sense that some of those stem cell populations would be very permissive to transformation. And there are many reasons for that. But you can see here that there are many researchers, including myself in the Dirks lab, that really thought that activated developmental signaling pathways may actually represent the fact that normal stem cells acquire mutations or other changes that could transform them into a cancer stem cell population. And in fact, this is essential to the evasion of therapy because these stem cells have a lot of different mechanisms that allow them to evade therapy. They have ABC transporters on their cell surface that allow them to efflux chemotherapy out of them so that the chemo can't kill them. They have enhanced mechanisms of DNA repair so they can become radio-resistant. And they have a habit of hiding out in the quiescent G0 phase of the cell cycle so that they can avoid systemic therapies that target actively proliferating populations. So all of those features really empower stem cells to be able to evade therapy. And when we perform those functional assays I told you about, we actually find that it really is those brain tumor initiating cell populations that become enriched after therapy. So in surgery, we use the term debulking all the time. We say, oh, I debulk the tumor. That means we took away the mass effect of it. We left some of it behind because it was stuck to dangerous, important structures, but debulking is good. And in the same way, I think chemo and radiation really only debulk some tumors. They take away 90% of the rapidly proliferating cells, and they leave behind a reservoir of cancer stem cells that are able to evade those therapies that then sort of regroup, just like soldiers, the only soldiers that aren't injured in an advancing front, and then they're ready to regenerate the tumor. Knowing this, we really do want to think about finding mechanisms of stem cell self-renewal to target when we develop those targeted therapies. So here you can see, and this is really frightening. Remember I told you that David gathered all of those samples at every stage in the sample. So here's engraftment, here's after radiation, after chemoradiotherapy, and at relapse. And the frightening thing is the sphere formation assay, which is a parallel assay to estimate self-renewal. You can see how frightening it is that as the cells go through the cycle of treatment, there's an increasing self-renewal capacity. And in fact, by the time you finish the relapse, you can see the frequency of the stem cell population has frighteningly gone up terribly, indicating that those stem cells are really taking over that population. And that is why they're able to establish the relapse tumor. Just last year, my friend Michael Taylor that I mentioned before, who spent a lot of time during his PhD arguing with me that it's useless to study cancer stem cells because they don't exist and cancer was a genetic and a genomic disease. And you know, there was no point in that I should really just join him in doing genomics, published some papers that suggest that in fact, the cell of origin of medulloblastoma is a normal neural stem cell. And so this really does bolster our entire comparative biology approach, even more so than our own earlier publications. And so this theme really running throughout everything suggests that the developmental neurobiology paradigm used to study this brain tumor is very, very appropriate. Here you can see our normal human fetal neural stem cells that we grow in the lab. This is our normal control cell population, and we profile all of the patient-derived medulloblastomas against this control cell population. So the other thing that we've been studying over time is BMI1, and it's probably no surprise. A lot of you probably know this gene is a chromatin modifier, an epigenetic regulator, but I know it as a master regulator of stem cell self-renewal. And so to me, studying this pathway in particular is another theme that has come throughout our work, because we have published many papers suggesting that that stem cell population that is enriching at relapse very highly expresses this gene BMI1, and at the protein level as well. And so we've invested a lot of time in wondering whether this particular gene and pathway and protein is targetable. And in fact, there is a company called PTC Therapeutics that makes a drug, a small molecule inhibitor. They specialize actually in orphan diseases, rare diseases, and they develop drugs for a lot of neurodegenerative diseases. But they have a small cancer program, and they had this drug, a chromatin modifier, that we found in the end actually binds beta tubulin, but indirectly has a significantly huge impact in reducing the expression levels of BMI1 greatly in medulloblastoma. And in fact, there's a gene dosage effect where you see BMI1 is expressed in normal neural stem cells, but it's expressed at a much higher level in medulloblastoma, such that there actually is a therapeutic window where you can titrate this drug so that you can hit the medulloblastoma stem cells and kill them while sparing the normal neural stem cell population. And you can see here that, remember earlier I mentioned that we like this model because you can layer drugs on. And so here is David experimenting, adding the BMI1 inhibitor onto standard of care. So what we found is that, yes, I know I have to get a better picture of Jason. He looks really depressed there. That's his picture on his website though, but I will get a better picture of him. Yeah, he looks grim. Yeah. So we realized when we treat medulloblastoma with both standard of care and the BMI1 inhibitor, we do drive the therapeutic bottleneck to an even smaller population of clones, but we're not yet solving the tumor or resolving the tumor. So we really want to understand what those treatment refractory populations are and what they do. And that's why we employed DNA barcoding as well as genome-wide CRISPR screening to really try to identify, is there a clonal population that escapes after the BMI1 inhibitor? And if so, how do we target that clonal population? And is there another drug that perhaps could work synergistically with the BMI1 inhibitor? And so this is just a schema of how we did the clonal dynamic experiment. And you can see we dropped that beautiful barcode library with 2 million independent barcodes onto our human medulloblastoma cells, which we then engrafted into mice. And the mice were then treated in all of the different arms. And we are able to then really quantify that there is a successful extraction and identification of the barcoded cells, and we can recover those barcodes. And not surprisingly, you can see the engraftment rate tumor initiation rate is about 10%, even with the barcodes, which is very standard in the literature. So all of this is great. And you can see here that when you treat with standard of care, there's quite a few clones that escape therapy. And this is the clones we recovered from the spine on the y-axis, the clones we recovered from the brain on the x-axis. And you can see when you combine standard of care with the BMI1 inhibitor, you're narrowing that bottleneck down to, there is a single clone we found that is driving spine and brain relapse. So this gives you hope. This is a disease that you can whittle down to homogeneity. This is a disease that you can drive to a certain point where hopefully there's only a single other set of signaling pathways that you could target to kill those remaining clones. So this gave us a lot of hope that we could work with this. So I mentioned to you before that a lot of our data sets can be repurposed and reused for other projects. And I mentioned to you now that we did CRISPR screening. Well, isn't that great? I'm sure that there's other people in the lab that would want to look at that CRISPR screening data. And so what you can see here is that step one shows the schematic of how we did a CRISPR gene, in this case, not for sensitizers or resistors, but, I want to get rid of that there. Not for sensitizers and resistors, but rather just to see what are the essential drivers of human medulloblastoma growth. And step two shows the filter. So after we removed all of the pan-essential genes, what we did is we compared to data sets of CRISPR screens of normal neural stem cells, and we filtered out the genes that were essential to normal neural stem cells to really find those context-specific essential genes that are only important for medulloblastoma survival. And you can see here that many of the top group 3 MYC amplified, the worst type of medulloblastoma, many of those genes are actually enzymes that are active in metabolic pathways. And this is very intriguing to us. A lot of these metabolic pathways have been implicated before, but really haven't been studied extensively in medulloblastoma. So here you can see a lot of those pathways. And we really developed an interest in pyrimidine biosynthesis based on this CRISPR results. And so we went on to do untargeted liquid chromatography mass spec metabolomic profiling of our group 3 lines with our collaborator at the Donnelly Center, Rafa Montenegro Burke, who you remember from being our very first lecturer in this CDCR lecture series. And you can see that there's an increased abundance of de novo sugar monophosphates in medulloblastoma cells, which you can see in the middle of the heat map here, right? And you can see that the purine and the pyrimidine salvage metabolites are actually significantly depleted in tumor cells relative to neural stem cells, which you can see at the top here, right? So blue is low and red is high. And so what this says is that Group 3 MYC-amplified medulloblastoma cells sustain their needs for purine and pyrimidine metabolites by relying on de novo biosynthesis, not the salvage pathway. And medulloblastoma cells have a vulnerability because the neural stem cells can still retain the use of their salvage pathway, and they're fine if you deplete them of pyrimidine. But this seems to be an essential vulnerability that we found in medulloblastoma. And in fact, we confirmed that. And we were able to see that loss of DHODH, if we knock it out with CRISPR, this actually selectively targets the medulloblastoma stem cells and spares the normal neural stem cells. And this is all work done by Will Gwynn in my lab, who is asking these very important questions. And he went on to actually show that loss of DHODH by CRISPR actually does decrease tumor engraftment in vivo. So then we went on to find small molecule inhibitors to see, can we recapitulate the genetic knockout with a pharmacologic approach? And there are available DHODH inhibitors. This one was from Bayer. And so we found the small molecule inhibitor, and it did indeed demonstrate very potent, and again, as predicted by our screens and validating, I think, our comparative biology approach. You can see here that there's a very distinct sparing of the normal neural stem cells, but a very clear targeting of all of the group 3 medulloblastoma cells with this DHODH inhibitor. And again, that is because the normal neural stem cells can still use their salvage pathway. So we went on then to show that these blood-brain barrier permeable inhibitors really do show great efficacy in our models and our PDX models of group 3 medulloblastoma. And you can see here what good effect we were able to show. So here you can see that really we wanted to try to understand better how the loss of DHODH impacts de novo pyrimidine biosynthesis. And so here you can see it's a very early step in pyrimidine biosynthesis. The conversion of dihydroorotic acid to orotic acid is regulated by DHODH. And this is where the Bayer drug actually works. And I want you to pay particular attention here to this oncometabolite UDP-GLICNAC, because it becomes essential to our greater understanding of the mechanism of what's happening with the DHODH inhibitor. So we were able to show by all of our metabolomic studies that there really is a really good increase in, or rather decrease of UDP-GLICNAC when you treat with the DHODH inhibitor and essentially arrest the pyrimidine biosynthesis cycle at this stage. Okay, so why is this important? And another question that came up is, why is this drug so effective, particularly in MYC-amplified medulloblastoma? Because Will had tried it in other subtypes of medulloblastoma. They're called group 2 and group 4. Really not as much efficacy seen there. So we began to question whether this mechanism was actually MYC dependent. And very, very fascinatingly, we were able to show that when you treat with the Bayer compound, the DHODH inhibitor, and you get a decrease in UDP-GLICNAC, this is normally, what happens is this oncometabolite normally O-glicnacylates MYC and allows it to stay and not be degraded. But when you decrease that pool of UDP-GLICNAC and you no longer have O-glicnacylation of c-MYC, it gets degraded. And so this is the incredible potency of this drug, that MYC, of course, is thought to be undruggable, but we're actually able to show a mechanism that implicates that MYC is degraded. And so this was really exciting because it brings up the fact that there are actually many other c-MYC amplified tumors in pediatrics that could be targeted with the same DHODH inhibitor. And finally, we were able to show that the DHODH inhibitor is on target because uridine can completely rescue the supplementation, when supplemented, can completely rescue medulloblastoma cells. And so if we circle back and we look at this, some pathways that are upregulated from our therapy-adapted models of recurrence, so such as c-MYC or OXPHOS or mTOR, or you can see here, are actually downregulated by DHODH inhibition. So remember all of that genomic data that David and others have collected at the beginning. We can actually go and look and recognize that some of the hallmarks of recurrence are actually downregulated by the DHODH inhibitor. And this bodes very well for looking at the fact that all of these pathways that are active in relapse, including mTOR, can actually be downregulated by DHODH inhibition. And then, I wanted to share with you that there was a very interesting gene that we pulled out when we actually looked at the differentially expressed genes in DHODH knockout cells. And you can see here, all the way on the side here, you can see here that EGR1 is a gene that really, really comes out. And this is so fascinating because in the central nervous system, we have a response called the immediate early gene response. Immediate early genes are critical regulators of environmental and gene environment interactions that allow environmental stimuli to trigger a very fast and neuroadaptive response to neuronal activity and plasticity. And what's really fascinating is that these transcription factors, such as EGR1, can elicit a rapid and dynamic response to neuronal activity, followed by a second wave of transcriptional regulation that encodes the adaptations at a synaptic and neuronal level. And so here you can see the role of EGR1 in cancer, and I'll switch to here. So here you can see the DHODH inhibition induces a very important metabolic stress response, and we think that this is mediated through EGR1. So here you can see that regulation of EGR1 regulating postnatal neuroplasticity via an epigenetic mechanism. And this epigenetic mechanism was published in a paper on normal neural stem cells showing that EGR1 recruits TET1 to actually shape the brain methylome during development and upon neuronal activity. And you can see here that the EGR1 target gene becomes differentially methylated if we actually, here we are, treat with the BAIR inhibitor. So if you see this heat map in the middle, here you can see that the EGR1 target gene is becoming differentially methylated when we treat the medulloblastoma cells with the BAIR inhibitor. And this is an agreement with changes in ERK signaling and the downstream CGN signaling as well. And this suggests that there's a functional enrichment of the EGR1 regulon-associated genes when we treat with the DHODH inhibitor. And so it's really fascinating that methylation profiling validated this finding in our own work. When we went back with single cell profiling to look at the EGR1 stress response, we can actually see that c-MYC-driven phenotypes are anti-correlated with the EGR1 regulon. And so this significant correlation suggests that EGR1 is part of the mechanism along with MYC, and really was incredibly fascinating because this mechanism brings us back to normal neurodevelopment and further strengthens that whole concept that perhaps this pediatric cancer really is development gone awry. So in summary here, you can see that DHODH catalyzes normal de novo pyrimidine biosynthesis. And really, it's now emerged not only with our work, but at the same time as our paper was published, there were two other papers that established the role of the DHODH inhibitor in interfering with pyrimidine biosynthesis, not only in two other brain tumors, not only in IDH mutant glioma, but also with DIPG or diffuse midline glioma, which is another very deadly brain tumor. And we think this acts on target in medulloblastoma, unlike the other settings where c-MYC doesn't play a role, but with us, we think it's reducing protein olignacolation, which leads to c-MYC degradation. And finally, this fascinating thing of pyrimidine starvation involving this, evoking this metabolic stress response, and we think this is regulated by EGR1. So the really interesting thing about this work is that using that comparative biology approach, we actually found a cancer-selective therapeutic strategy that spares normal neural stem cells, which are so important in this population of children we're treating who are 3 to 5 years old, and that perhaps there are targetable metabolic dependencies in relapsed medulloblastoma. And so, I mean, this work was really exciting because the 3 papers coming out together really presented a very strong argument to take this drug to clinic and to perhaps consider something like a molecular basket trial where you could enroll children across all kinds of different MYC-amplified cancers, those with diffuse midline glioma or DIPG, those even other children with high-grade gliomas, IDH mutant gliomas. And you could actually consider treating them with DHODH inhibitors if you could also develop some good metabolic biomarkers to understand how to stratify children for such a trial. And so the great thing is the authors of all of these papers got together with some drug developers to explore exactly how to make this happen. And so this sort of exploitation of de novo pyrimidine biosynthesis as a key vulnerability across multiple brain tumors has been a really exciting application of our metabolic investigations. So here you can see we just did a little bit of work into trying to develop some of those biomarkers if such a clinical trial were to be started. And we see that if you overlay pyrimidine and c-MYC transcriptomic and proteomic signatures, you really can find that there's perhaps a strong subgroup specificity for pyrimidine metabolism. And you wonder if there could actually be, along with the molecular, could there also be a new metabolic classification of medulloblastoma that could have some bearing. So I will finish that story there because I think it really is one of the nicest paradigms illustrating how we were able to mine the data generated from our models and how we were able to use our comparative biology approach to uncover new metabolic vulnerabilities. But I want to tell you a little bit about one other branch of our lab as well. We have a branch of our lab that is very, very focused at trying to apply some of the novel immunotherapies that we've been able to develop for GBM also to this childhood brain tumor. And this is in keeping with what's happening in the world of pediatric cancer, where at Seattle Children's Hospital, they've been really front runners in terms of developing new CAR-T approaches for children, and also at Stanford University, where these CAR-Ts are delivered local regionally. So in the first slide I shared with you guys about the patients, you could see that the CSF spaces are there. And very often, as surgeons, will put a reservoir into those CSF spaces, and this presents a perfect way to deliver new therapies directly to the brain tumor. You don't even have to think about the blood-brain barrier and how you get across it. You can just bypass it. And this is a very sensible way to also think about developing a biomarker strategy, because if you're treating and delivering into the CSF, you can also sample the CSF. And by sampling the CSF, multiply. Again, you don't ever want a single snapshot of anything. You want to be able to profile something through space and time. So being able to take multiple samples of CSF over time can give you an idea of how a child is responding to a therapy, can give you an idea of the inflammatory cytokines that are released and how a child may be responding, and maybe gives you a heads up to treat a child before they develop serious complications. There's so much information that you can gather, which is anticipatory in nature. And the best thing that we could possibly do is develop predictive biomarkers alongside our new therapies. So that's another thing we really have our eye on in SingLab. So now I want to share with you some of the work from our wonderful MD PhD student, Eugen, who I think most of you know because she doesn't hide under a bushel or anything. So Eugen made great use of the databases built by several other students that left as she started in the lab. So she went and exploited some of the data generated by David Bakshinyan that I shared with you earlier, as well as some profiling done by another very talented MD PhD, Michelle Cometa-Smith. So you can see here that Eugen was really interested in looking for new CAR-T targets that are specifically expressed in recurrent medulloblastoma and perhaps not your usual canonical CAR-T targets. And in doing that, she wanted to broaden her search to look for more than just your usual, hey, I'm on the surfaceome, I can be targeted with a CAR. She took a bit of a broader look at things. And so here you can see that she filtered out all of these proteins that were enriched at recurrence post-chemoradiation. And here are the methods that she used to go down and filter down to some potential CAR-T targets. And you can see here that she found 3 that by all of her metrics, and one of her criteria was, of course, that it shouldn't be highly expressed on normal cells, it should be more expressed on cancer. She emerged with some of these proteins here. And what is very, very interesting is, I'll just skip on to the next one. This is just another strata of how she stratified everything. But you notice that one of those proteins was called LBR, and laminin receptor beta is actually a nuclear envelope protein. And this is very perplexing because you wouldn't necessarily expect to see that a nuclear envelope protein would have a very important role in medulloblastoma genesis or anything. But here you can see the 2 membranes of the nuclear envelope. They're separated by the luminal space and continuous with the bulk endoplasmic reticulum network. And the inner membrane is characterized by a set of integral membrane proteins that connect the nuclear envelope to chromatin. And here you can see that LBR actually is essential for cholesterol biosynthesis, and it anchors the lamina and heterochromatin to the inner nuclear membrane. And you can see here that there are some other essential nuclear membrane proteins. There's LAP2 beta and there's emrin that are also similar to LBR. And what Eugen found is that LBR was highly enriched only in MYC-driven group 3 medulloblastoma compared to normal neural stem cells. And in fact, she could see something very weird. And this first observation, we questioned heavily because we just didn't believe it, to be honest. What we noticed is that Eugen was showing that LBR was not expressed on the nuclear envelope. In fact, it was on the plasma membrane. And we're like, oh, that must be a mistake. That's not real, you know? But you can see here that, you know, she actually did manage to prove that it was actually on the plasma membrane. And you can see it with your own eyes there on the plasma membrane. And it appears, and our working hypothesis at this point, is that LBR was somehow mislocalized to the plasma membrane only in the setting of group 3 medulloblastoma. And using some help from Jorge, a very talented postdoctoral fellow in the Crenciuti lab at St. Jude's, one of our collaborators, you can see here that we were able to co-localize the LBR with a resident plasma membrane marker and show that it seems like at any given time, about 15% of the total LBR is at the cell surface. And this is really fascinating because that is not where it's supposed to be, begging so many questions about how is it being mislocalized. The very exciting thing is no matter how many normal tissues we looked at, we could never find a normal tissue where LBR misbehaved. LBR always remained where it was supposed to in every normal tissue. It stays on its nice protected nuclear envelope where no CAR T cell could presumably ever find it, right? But when something is presenting on the plasma membrane and it's there on the surface, it's potentially targetable. So wouldn't it be great if there was something that was only expressed on group 3 medullo at the surface, but in every other cell, it's on the nuclear envelope. And in fact, even within all the medulloblastoma subgroups, you can see the surface localization seems to be fairly specific only to MYC-driven group 3. And in fact, LBR protein expression, much like the patterns of the brain tumor initiating cells I showed you earlier, tends to enrich and become more highly expressed as we go through the therapy and at recurrence. And so I'm going to leave you with this very intriguing story at this point, because I'm pretty sure that Yujin wants to give a seminar to tell you about all of the rest of how she's done it. But this is really what I've now challenged for Yujin with for her PhD thesis. I've said to her, okay, so this seems to mislocalize to plasma membrane only in hyperproliferative medulloblastoma stem cells that escape therapy. So now it's Yujin's job to figure out what and how and why. And so we're really now searching for the mechanism of this apparent mislocalization of LBR to the cell surface, to the plasma membrane. Why is it only happening in MYC-driven group 3 medulloblastoma? And can we develop a targeting strategy if all of this is true? And so the concept would be to develop an scFv against LBR, or perhaps against multiple nuclear envelope proteins to increase your coverage, because remember, it's only 15% of cells at any time that have it at the surface. So what if you could target multiple nuclear envelope proteins, maybe with multiple pooled CAR Ts or with a poly-targeting CAR T? But it really presents a lot of interesting ideas that we've shared with Kevin Henry at NRC, who you remember was also a speaker in our series. So this is currently something that I'll leave to Yujin to update you about, but I have charged her with showing the real proof in real time. So I've said to her, what would be really lovely is with dynamic microscopy, could we somehow label LBR and visualize it, perhaps trafficking in little extracellular vesicles as it mislocalizes, incorporates into the plasma membrane. If you show it to me in real time, I'll believe it. I told her it's something like catching Trump on tape, you know, like you need evidence, right? So here you can see that she's enlisted the help of Jorge, and Yujin has successfully managed to endotag LBR such that hopefully we really will, maybe with a snap tag or something, we'll actually be able to visualize the localization to the cell surface. So stay tuned. I will leave those beautiful videos for Yujin to share with you one day soon. So this really represents a paradigm shift. Yujin's work, it's very powerful. Yujin was fortunate to be able to go to Stanford and visit Crystal Makel's lab to learn more about CAR T-cell engineering. And there she shared this work with them. And they really identified, like, this is amazing, because in the normal schema of how you look for CAR T-cell targets, you would never have ever found a nuclear envelope protein in any realm. And so our discovery pipeline actually facilitated this discovery, which is somewhat serendipitous and unusual. But if it really does bear out, then it's possible that this could actually change the way that we think about CAR T-cell targeting. And perhaps we don't need to just think about the previous schema in terms of finding our new targets. And perhaps this is a way to also find safer targets, because they might be targets that are only relevant to certain context-specific cancers. So I'll finish last few minutes to tell you a little bit, when sharing this from one of Crystal's beautiful papers, that really to get ahead of medulloblastoma, just like I recognized when I was in neurosurgery, that I can study everything there is known to neurosurgery, and I'm not going to durably cure a medulloblastoma. I can take out a medulloblastoma and make the child feel a lot better, and in a small percentage of patients, surgery alone can cure. But without proper understanding of relapse and development of further therapies, I'm not going to do it alone. And so you have to collaborate with other people. And what we've realized too is that we know a lot about medulloblastoma targeting, but it's medulloblastoma biology, but it's really more to CAR T experts to help us to discover what are the appropriate therapeutic strategies that we can tailor to the specific biology that we're seeing. So if we see something that's dynamic, how do we target it? If we see something that has a low expression level, how do we target it? right? These are problems that people like CAR-T engineers can solve. And so working with people in Mako Lab really enables us to employ all of these beautiful new technological approaches that they've developed to target something that may have a low density at the cell surface. And what if we're targeting LBR, but it's only on 15% of cells, and it's really a few receptors per cell, and it might not be enough to activate and kill those cells. So we have to find ways to engineer. And so this is why we've been lucky to collaborate with Crystal's lab in looking at an oncofetal antigen that they've been very, very interested in, and that they've studied in the setting of neuroblastoma, which is GPC2. So this is a heparin sulfate proteoglycan, and it has established signaling connections to many known developmental pathways, including Wnt and hedgehog and BMPs. And they have already proven that it's an immunotherapeutic target in neuroblastoma. And they published this beautiful Cancer Cell paper where they actually added c-Jun to really hone the approach of targeting these low antigen density targets. And they said to us, why don't we see if this also works in medulloblastoma? And what you can see here is that GPC2 actually is highly enriched also in medulloblastoma, and not as enriched in the brain of children who are more developed. And you can see here that when we experimented with their c-Jun co-expressing GPC2 CAR-T, we actually saw not only good results, but even that a certain cohort of mice were cured. And so this was really remarkable. With this very, very aggressive tumor, when you just implant this tumor and Manoj images the mice, the untreated die in 3 weeks. So to see some of the mice remaining tumor-free for more than a year was quite remarkable. So here you can see this is a nanoluciferase imaging that's built into the CAR-T, so it enables us to actually track the CAR-Ts and see where they're going. And you can see that c-Jun really enhances the CAR-T expression in vivo. So now I've sort of shared with you all of these different comparative biology approaches that we've used, and how we've sort of landed on finding new metabologenomic vulnerabilities, specifically in group 3 recurrent medulloblastoma. And all of these models have really helped to leverage us towards this goal. And we really have found that perhaps going after pyrimidine metabolism is a really cancer-specific and essential vulnerability. But the problem with targeting metabolism is that those things, unlike Eugen's mislocalized LBR, are all inside the cell, and they're not expressing at the surface, and they're not presentable as CAR-T targets, you would think, until we saw the work of Dr. John Maris and Dr. Mark Yamarkovich, who did this brilliant, brilliant work where they discovered that actually you can have a cross-HLA targeting of intracellular oncoproteins with these new peptide-centric CAR-Ts. So what they did is they looked at the immunopeptidome of neuroblastoma, and they identified that there was a certain class of HLA-presented oncopeptides, and they developed a counterpanning technology and developed these peptide-centric chimeric antigen receptors that can target non-immunogenic intracellular oncoproteins. And in fact, this paper was a landmark paper because they discovered that you could actually break HLA restriction. And the CAR-T cells that they developed against FOX2B actually specifically killed neuroblastoma cells, and they showed tumor regression in vivo. So this is the first proof of principle that you could target an intracellular antigen using a peptide-centric approach. And so we really thought this might be a good thing, given that so many of our hits, remember the hits in the original pipeline that Eugen had elucidated, she actually ruled out all of the intracellular proteins, thinking, well, we can't target those with CARs. So after we started collaborating with John Maris and his colleagues, Eugen promptly put all the intracellular targets back into our pipeline. So you can see here now that everything that we're mapping in terms of the metabolome is potentially targetable with these new CAR-Ts. But we really do want to understand better, what would be the mechanism of having metabolites presented at the cell surface? And this is where there's a new emerging field of immunopeptidomics and immunometabolomics that we've been very, very lucky to explore, along with our colleagues, John, Mark, and very talented Nick Sorgakis. And this is becoming the project of our postdoctoral fellow on the Medullo team, Lara Escudero. So the non-classical MHC-related protein MR1 typically, and this is wonderful for all my infectious diseases colleagues, finally I have something that I can speak your language about. So normally MR1 displays antigenic bacterial riboflavin and folate-derived metabolites to mates or mucosal-associated invariant cells. And in humans, these intermediates are recognized as non-self. And so intriguingly, MR1 has also been shown to display cancer-specific antigens at the cell surface. And that allows MR1 restricted T cells to eliminate cancer cells as well as bacteria. So just another example of how infectious diseases related approaches can always be extrapolated to cancer. And that's a conversation I've been having with my new colleague, John Stokes, lately. So inside the endoplasmic reticulum, MR1 is stabilized in a ligand receptive conformation by chaperone proteins, tap binding proteins. And now we can take those metabolic vulnerabilities that we identified in pyrimidine biosynthesis in MYC-driven medulloblastoma, and with our colleagues, we've already identified that some of the putative MR1 ligands that we discovered are displayed in group 3 medulloblastoma. And most intriguingly, and this data is so exciting, we actually found that UMP is actually one of those MR1 metabolites. So something we discovered as being a byproduct in pyrimidine biosynthesis is actually an MR1 ligand in medulloblastoma. And this is really, really exciting because it's actually been validated. Our colleagues used a computational approach to vet the structure of those oncometabolites against the structure of known MR1 bound ligands like folate. And then they actually were able to identify UMP as indeed a strong ligand. And then they confirmed that by doing in vitro binding kinetic experiments. And they actually were able to show here's UMP in D, and it's bound in the MR1 pocket. So this was so exciting to us. And the nice thing about this is that now we can actually think about developing not only peptide-centric CAR-Ts, but maybe metabolite-centric CAR-Ts. This is all a little bit still forward-thinking and something that's going to take a lot of time to develop, but this is a very exciting project that we think Laura will really be poised to work on. And if you think about metabolite display, well, there's also something to be said about lipid display. So going back to Eugen's project, we kind of speculated about whether lipidomics would help us understand how LBR1 could be mislocalized to plasma membrane, because perhaps there's a differential lipid composition of the cancer plasma membrane compared to the normal cell plasma membrane that could somehow contribute to this mislocalization. And so we've run a lot of lipidomics, and it turns out that a lot of lipids can also be displayed by CD1 on the cell surface, making potentially some of those abnormal lipids or ultra-high chain fatty acids that we've only found in medulloblastoma that don't exist in the normal stem cells. Perhaps those could also be targetable by the CD1 display mechanism. So there could even be lipid-centric CAR-Ts that we could develop in the future. So this project is really taking us into very exciting new directions. I'm so glad that Greg Steinberg moved into the office next door to mine because I have a lot to learn about metabolism, Greg. So I'll finish on this final slide. Wow, I'm never on time. You guys, look at that. So I want to finish on this final slide and tell you guys that we've been really lucky again to work with very talented people who have all the skill sets that we don't have to engineer CAR T cells. Because as Jonathan Bramson, who's not with us today, unfortunately, but often tells me, he likes to remind me frequently that I'm not an immunologist. I know, Bramson, I'm not an immunologist. It's very important to recognize your limitations. And that's where you reach out and collaborate. When you realize where your limit ends of your knowledge, that's when you step out and find either a way to augment your knowledge or find someone to work with who has that knowledge. So we've been very lucky to work, not only on the left, you can see that we're part of the Cure Group for Medulloblastoma Consortium. Stefan Custer's in my lab, who can tell you his story another day, is working to try to develop new therapeutics for this other class of treatment refractory medulloblastoma. We've been lucky to work with some of the top medullo scientists in the world, led by Dr. Roger Packer. And this is a huge deal for me because Roger Packer is one of the top pediatric oncologists in the world. He has led almost every pediatric cancer trial that you can imagine for the past 35 years. And he just has so much knowledge and so much insight. And whenever anything that comes out of his mouth just yields a huge amount of knowledge. And so it's great to work with a team like that to try to develop new therapies. And then on the right, we've been very lucky to join the research collaborative, which we're calling at the moment MedulloCAR. We're applying for a very large St. Jude's internal grant to be able to put together all of our knowledge to develop new CAR T therapy approaches for both medulloblastoma and neuroblastoma, as well as DIPG. So I will finish there to thank everyone in the lab. Everyone in the lab works together incredibly well. So even though there's 3 teams, there's so much crosstalk and support between the teams. But this small but mighty team of medulloblastoma scientists who recently welcomed Erica, who's sitting here and whose photo I don't have yet, and Sean, I need your photo for Team GBM. So, so you can see this group of people have done a huge amount of work. Whenever I go and present this work, it's a huge program. It's done by a small group of very hardworking people, and I really commend them on the incredible discoveries they've made. So thanks to all our funding partners, and I would love to take questions. Supposed to come back up now, Rulo, and take questions.
It's no virtual.
I don't know.
Yes.
And then, for example, the two from the example you showed today, also related to that is that even recurrent medulloblastoma from the same group of primary tumors, do you think it's a more similar type of disease, or there could be still different subtypes of the other?
Yeah, that's an excellent question, Hong. So unlike GBM, which you and I work on together, it seems like in GBM that every patient recurs differently. And it seems like we almost need to develop like a personalized biomarker-driven therapy that's different for every GBM patient. So I almost feel like after the bottleneck, it's divergent and it opens up again with GBM, right? But I feel like with medulloblastoma, through the bottleneck, it narrows, and it narrows to a point where there's only a few signaling pathways that all recurrent medulloblastomas rely on. So this is awesome, right? Because all of the diversity actually narrows down to almost a homogeneity, near homogeneity. And in fact, we think some of those signaling pathways involve insulin receptor, insulin growth factor receptor signaling, PI3 kinase signaling. Those signals seem to be active in every recurrent medulloblastoma by multiple groups findings, Michael Taylor and others, and us as well. And so what that leads me to think is something that was validated by Mike, which is that when he compared all of the primary recurrent medullo pairs to each other, they are more different than all of the recurrent medulloblastomas are to each other. So in other words, all of the recurrent medulloblastomas from all different patients are more similar to each other genomically than the same primary recurrent from the matched pair. So what that's telling me is the word I was looking for, sorry, GBM is divergent and medulloblastoma is convergent. And so I think that it's all converging to similar signaling pathways. And that's why at the beginning of the talk, I said, I'm hopeful that we can even cure relapsed medulloblastoma in the next 2 decades, because we're sensing this convergence to very minimal signaling pathways that are potentially targetable. So I actually, on my clinical side, have been talking to all my clinical oncology colleagues from the Group 4 Medulloblastoma Consortium. We're just trying to source every PI3 kinase inhibitor we can find. So now I've learned about all of them, paxilosib, buparlisib, there's like millions of them. We're trying to figure out which is the best one to take to trial and which is the most efficacious, because that seems to be one of those convergent signaling pathways at recurrence. So I have a little more hope. There's a lot more work to do for GBM, which is why it's good you're young.
Any other questions? I was quickly curious about the CAR T cell that recognizes the heparin sulfate. That's an antibody that's been engineered, or a single chain that recognizes a particular?
Yeah, that's based on an
glycosylation?
SCFD. Yeah.
There's heparin sulfate all over the place. There must be a unique
Oh, it's a very
file.
specific isoform of GPC2 that is only expressed in cancer.
Yeah.
And with oncofetal antigens, what you find is that they're expressed in the fetus at very high levels, but then they go away for all of adulthood. And then they're only re-expressed in cancer. So it should be a very specific target, we hope.
Yeah.
And it's already moving into trial for neuroblastoma and other cancers. So maybe medulloblastoma soon. Tobias.
I have a question about the mood-driven subgroup of people that it looks like that there are some very specific features, like the mislocalization of the DHODH certain metabolic pathways that they rely on, or that they for negative vulnerability. Is this something similar? It's also from the other mid-driven tumors like lymphoma or others that are mid-driven.
Yeah, that's a great question, Tobias. That's exactly what we're thinking, because much as the recurrent medullos all act like each other rather than the primary and recurrence from the same patients, it seems like this MYC-driven class of medulloblastoma is in a class of its own. It doesn't even behave like the other medulloblastomas, and I sense it's going to behave more like the other MYC-driven cancers, which really puts an argument forward for molecular basket trials. I'm not sure we should be moving forward anymore with strategies where we say, okay, all the medulloblastomas enroll in this trial. We should probably say, all the, UMP low pooled MYC expressing tumors come to this trial and get treated with this drug, right? I think I still am investigating this because our paper was only November, but I'm working with Daphne Haas-Kogan at Harvard, who published the DIPG paper. And we're trying to figure out and bring in neuroblastoma colleague, Burkitt's lymphoma colleagues to try to figure out if we should be running this trial in MYC amplified tumors and just enroll all of them rather than just brain tumors. So I think it's a very valid question. I think we have more mechanistic insight from this paper about the mechanism of action involving MYC degradation, but we still don't know all the answers. So.
Okay, doesn't look like there's any other questions. So join me in thanking Dr. Singh for a fantastic.